I'm glad to continue after Petra's talk and go exactly into the direction of how we can bridge the scales between the molecular scale and the large scale of brain simulations. And I don't have to explain to all of you how brain simulation works. So we typically use a skeleton of connectivity and bring it to life by local dynamic models. And all this is then fitted by using typically functional data. And what I want to focus in this talk today is how we can think a bit outside the box and implement information beyond just the classical imaging, and especially from the molecular scale, because as we all know, in general, although it's a simplification, whole brain model captures the brain starting from the large scale, but also on the mesoscale, like for example, neural circuits, but down to synapses and synaptic features, and indirectly also by molecules. So why do we need this bridge from the subcellular level up to the brain? Because we want clinical applications. So I'm working in research, but also in clinical neurology. And for me personally, it's always important that our research comes into a clinical application. And the thing is, neurological diseases typically have a surrogate on the molecular level because the mechanisms are usually on the level of proteins, of genes, and therefore it's important to link this to the simulations. And in this talk, I will show you some ideas and approaches how this can be done from recent works and also from ongoing works. I start with this now even a bit older study, which I did for my PhD thesis, where we used a group of Alzheimer's disease patients and implemented molecular information from PET imaging. So we took data from ADNI and implemented amyloid PET burden. So we had an information for each region, how much of this amyloid beta, one of the two hallmark proteins of Alzheimer's disease, was in this region. And then we used a mechanism from the literature that it is known that amyloid beta typically affects inhibitory neurons and leads to a disinhibition. And this we implemented as a cause and effect function into the model. And by doing so, we were able to reproduce phenomena that are known from Alzheimer's disease, so typically a slowing with a shift from alpha activity to theta and delta activity. And we were also able to show that when we use this simulated local field potential, so basically artificial data, and plug it into a machine learning classification model, we can improve the diagnostic classification between Alzheimer's disease, healthy controls, and mild cognitive impairment, which is kind of an intermediate state. With the same model, we were also able to show how therapy works. So we used as an example memantine, one of the two groups of classic antidementive drugs. It's an NMDA antagonist, and it can improve memory function in Alzheimer's disease, but it had also been shown that it reverses some electrophysiological effects. Typically, the slowing that we see in EEG can be reversed by memantine. And when we applied it into the same model where we saw the slowing caused by the amyloid beta leading to disinhibition, leading to slowing, and we added virtual NMDA antagonism, then we also saw that this slowing disappeared again, resembling the pharmacodynamic effect of the drug. We were very happy also to see that in general, this particular mechanism in Alzheimer's was able to being reproduced in different studies. So in these two, we were involved. This one used the same model, so a Janssen-Ritten model, but not amyloid PET, but a model, a very complex model of how the proteins of amyloid and tau interact with each other and lead then to this disinhibition, showing the same relationship. And this work used a different neural mass model, so the balanced excitation inhibition model by Gustavo Deco, and fitted to functional MRI, the cause and effect function, again showing that this relationship between amyloid beta and inhibition is needed to maintain and to reproduce the patterns that we see in empirical data. And this one was that we were not involved, showed also that they actually used the same cause and effect function with a completely different thalamic model and also showed the slowing. So this is, I think, a nice example to show how a molecular mechanism can be implemented into a simulation. So as I said, as I showed, we can use these mechanistic simulation to improve diagnostics, to test out and understand virtual treatments, but mostly it's about the mechanisms inside. And I think this is also the most interesting, maybe also the most complex part of potential applications. But I think it's the one that might be the most powerful. And if we think of the brain as a mechanistic machine, then we can think about what we can do with these mechanisms in the area of brain simulation. And I will speak about how we can use data or information to constrain the simulations in a mechanistic way. I will also show how we can understand mechanisms better by this tool, and talk a bit about how we rebuild mechanisms in a brain simulation framework. So I start with the constraining, and of course, the most obvious thing to constrain a model is to use data. And there are, for example, databases like eBrains, where a lot of data from the molecular scale is available, for example, receptor densities, gene expression maps, and so on, that can be used as a constraint matrix in a similar way like I showed it first for the amyloid PET. But there are also a lot of processes in diseases where we don't have data available. We have information, like, for example, here, this is NeuroMMSig. It's a graph database showing how different entities interact in Alzheimer's. So the small nodes are proteins, genes, chemical compounds, but also processes. And they describe the relationship between these entities, typically derived from the literature, but then also curated and assembled from many different graph databases. And this is a very powerful tool because it tells us how mechanisms in diseases work, but typically we don't have the spatial information. So we don't know where exactly all these interactions happen in the brain, but this is crucial to implement it into the brain simulation. And therefore we came up with our own tool to map these things. It's still unpublished, unfortunately, but hopefully we will publish it soon. We use the concept of semantic mapping to create maps for things that we have not mapping data available yet. And for this, we used a software that our colleagues from the Fraunhofer Institute have already developed, Skyview, which is basically a advanced PubMed database. So you can search for a query and get results from scientific articles, but the articles itself were curated and especially with controlled vocabularies. So words in the articles are marked, for example, as genes or as proteins, but also as anatomical terms. And there we use the Uberon ontology describing all kinds of anatomical nomenclature and also the relationship in between the anatomical nomenclature terms. And as we have this information, we can extract statistics about this. So basically, we have a query. We search, for example, for memory in Alzheimer's disease, and then we get a list of several thousands of papers, and we can see which anatomical terms are more often in this query, in these results, than others. And what is missing is because then we just have a list of anatomical terms, so we have to map it onto the brain. And this was a manual work of anatomical curation. So we made a process where we, in a detailed procedure, mapped about 1,000 anatomical terms onto the three-dimensional space of the brain. And by this, we have a transformation matrix that can project this statistical coappearance in the scientific literature onto the brain, and therefore allowing us to map every possible semantic query onto a brain map. And we were able to show that this actually works very well with kind of all modalities, so ranging from MEG spectra to receptor densities to glucose metabolism or brain functions. They're typically captured quite well just because they're using the big amount of more than 30 million scientific articles. So we have data to constrain the model, but of course we want to understand then the mechanisms better, because this is how we can get really into clinical applications. And therefore I continue directly with the software that I just presented. So as we had it, we wanted to try it out if we can use it also to decode patterns that we know from empirical data. And here we used results from colleagues from us from Munich that have extracted patterns of volume changes that are predictive for the onset of psychosis and people at risk of developing psychosis. And we wanted to understand which genes and which proteins and which clusters are related to this to these patterns, because this is maybe then leading to a potential way to modulate it or stop it. And we mapped with the Stevie-based software all human genes from the human gene nomenclature, and then used the support vector regression to reconstruct the pattern. And this led to mainly 3 clusters explaining very well the volume change pattern. 2 were quite expected, which is neural growth and neurotransmitter homeostasis. But we also found the 3rd cluster about neuroimmunological interaction, mainly about tumor necrosis factor, which is very interesting because it's also something that we can use for potential treatments. And we found did a very similar thing with Alzheimer's disease. So we used the graph database that I showed in the beginning, NeuroMMSig, describing the interactions of proteins and other stuff inside of Alzheimer's. And we mapped all these components with our software. And then we extracted a core cycle just by selection criteria of the maps that contain the most information. And this led to this kind of vicious circle of Alzheimer's, showing like neuroinflammation, again, mainly by the central component of the tumor necrosis factor, can maintain a vicious circle leading to both amyloid beta and tau deposition. And we wanted to explore this more in a simulation framework. And so we used the cause and effect models that I showed in the beginning, again with ADNI data, but we used a subset of ADNI where there were immunological markers in CSF available, and actually also the TNF concentration in CSF. And we wanted to see, is there a coincidence between inflammation and hyperexcitation? And actually, this was the case in the group of mild cognitive impairment, and not in healthy controls, and also not in Alzheimer's disease patients, as you can see here. And this is very interesting because it goes along with a common hypothesis that the phase of excitotoxicity, so overwhelming excitation that leads to inflammatory reactions, is typically an early phenomenon in Alzheimer's disease, afterwards going over into a more neurodegenerative track that then leads more to lower excitation just because the neurons actually die. And because we wanted to understand this phenomenon even more, we also looked to other immunological molecules. And interesting, when we connect all these different immunological interleukins and so on to excitation in the Go network describing biological processes, most of them really go through this TNF, showing that actually our hypothesis makes sense. And when we compare the other immunological mediators that were available in the subset of ADNI, we see that also almost the strongest excitoinflammation, we call this, so coincidence of excitation and inflammation is actually led by TNF-alpha. So as I showed, this is again an example from Alzheimer's disease, how we can understand mechanisms even better with brain simulation. But of course, it's always important to rebuild the mechanisms then in a proper way, because that's actually the hard work, and where the small details are important. And one thing that we therefore addressed is that we wanted 2 things that we did not have available yet. First, we had a lot of very interesting molecular data or microscale data from mice, and it's of course it's possible to simulate also with mouse brains, but it's not done as often as with humans, I would say. And on the other hand, we also wanted to have a direct connection from many things like gene knockouts or protein simulations to the brain models. And in my view, the best and most direct connections is on the level of ion concentrations, because for example, when we have a simulation of what a knockout in a particular channel has of an effect, then often it affects the concentration of ions, typically sodium, potassium, and calcium. And therefore, we wanted to set up a model that captures all these, which is, for example, the Lata-Breakspear model inside a mouse framework, to be able to understand how these and to implement how changes in ion concentrations drive the dynamics of a mouse brain. And actually this worked, so we were able to reproduce functional connectivity based on local field potentials quite well. And we were also able to show that all ions, so calcium, sodium, and potassium, shift the dynamic state, typically in later Breakspear from the fixed point state and from the oscillatory state to the chaotic behavior. And interestingly, we also showed that this model reacts to stimulation, and the perturbability, so how strong it reacts to stimulation, can also be moderated by the calcium Nernst potential, allowing building a framework to explore how stimulation tasks might differ depending on, for example, channel mutations. And as I said, it's always important to rebuild these mechanisms in a proper way. Therefore, I come back to the NMDA antagonism. So I showed an earlier work where we showed how the memantine modeling could show phenomena like in BC and EG of Alzheimer's patients. And as always, as modeling, you need a model that is as simple as should be as simple as possible, but as complex as needed to maintain the phenomenon that you want to present. And when we want to build the whole effects of NMDA antagonism, we have to keep in mind that NMDA receptors are not only on excitatory neurons, they are also on inhibitory neurons, and there are complex effects because they work, they act anti-excitatory and pro-excitatory at the same time. And here, a PhD student, Jenny Them, was able to show that when we implement the effects of NMDA receptors on all three cell types of the Janssen-Ritt model, we are able to reproduce the exact path of what we see when we under a ketamine narcosis, so how the firing rate increases and alpha and theta band change between each other. And finally, I just want to close the loop because I talked a lot about how to implement and to plug in molecular information into the simulation. But the question was, can we also get an output on the molecular scale? Because typically the outputs are all on the electrophysiological scale except of Bolt. And inspired from the Bolt monitor, we were thinking of, can we also reproduce glucose PET as a very direct surrogate of energy metabolism. And for this, we employed a detailed microscale model of neurogliovascular ATP homeostasis, so describing how ATP, actually the intracellular correlate of glucose regarding the energy metabolism, is transferred from blood vessels to neurons and glia cells. And by fitting this model then to functional glucose PET data, we were able to explore the difference between the response function of Bolt and of this PET, which actually is very interesting because it's two methods that measure in general a similar thing, which is brain activity, but on a different time scale and also with a very different method. And we were able to reproduce the static and functional PET actually quite well, although it disappeared on the slide. You have to trust me here. And therefore, we will offer also then a second correlate of brain activity that can be used in brain simulation. And finally, I just want to conclude that because we were working so much on implementing mechanisms in different models and so on, we also thought of that this should be more standardized and more in a controlled way. And therefore, we built an ontology describing the different components of the virtual brain, so the neural mass models, the parameters, and so on, and how they interact. And this ontology is also linked to biological processes from the Go pathways that I showed before, and therefore can be used in future to justify mechanistic simulations a bit better by referring to an ontological framework. And with this, I end, because I think also the time is running. And thank you for your attention, and my colleagues, that especially those of whose work I presented in this talk.
Great.